This so I will present you George Tsirtsis, who came from the University of the Aegean. He's a professor of dynamics of coastal ecosystems there, and he's visiting Glasgow with a MASTS small grant. And I know that he has a slide where he's giving an overview of his research interests, so I don't want to delay his talk so that we have enough time at the end for any questions. So George, do you want to?
Thank you very much for the introduction. I will start with the title of my presentation, which is about the ecological variation of phytoplankton in the Aegean Sea in Greece, and some inferences about the predomination of neutral or niche processes in the structuring of phytoplankton communities. Just a few words about my research interests. First of all, the composition of phytoplankton and productivity in space and time is one of my research priorities, and the study of the role of scale. I have an indicative paper on this related to a downscaling marine modeling effort that we made in the past. The second point is the terrestrial inputs as a diversity sustaining mechanism for phytoplankton, and another paper. Third, trying to infer patterns from processes, which is very important in phytoplankton in the light of modern ecological theories, like the niche theory or the neutral theory, on which I will spend some time in the talk today. And finally, the limitations that they are imposed by simulation models or experiments, experimental systems, like mesocosms or microcosms, when trying to study various phytoplankton, various scientific questions related to phytoplankton. So, about my talk today, which, as I told before, is on the relative contribution of neutral and niche mechanisms on structuring phytoplankton in a large marine area like the Aegean Sea. Everything started from a project that we ran during the 3 years, which is the ECOGENE project. The title of the project is The Relative Role of Niche and Neutral Mechanisms in Controlling Phytoplankton, Genetic and Morphological Diversity. The underlying question, the question on which the main scientific question of the project was the relative role of those mechanisms, but we approached this scientific question using data originating from both molecular and morphological identification of phytoplankton. We used 3 approaches in sampling. First of all, we sampled 9 Aegean gulfs in July 2014. Those gulfs can be seen in this slide. There were 2 gulfs in the island of Lesbos here, 2 gulfs in the island of Limnos, and so on. So those 9 gulfs cover the whole area of the Aegean, and the sampling there was simultaneous. All gulfs were sampled during July 2014, trying to study only the spatial changes and trying to exclude possible temporal effects on the composition of phytoplankton. Secondly, we made some mesocosm experiments in a place where salt is produced, in a salt works, where there is a very interesting gradient in salinity. So we used 3 salinities and tried with a number of mesocosms inside these ponds of different salinity, and we made an experiment trying to see if dispersal between ponds of different salinities is important for the structuring of phytoplankton. The idea behind is to check if local processes related to the environmental conditions of each pond, of each salinity, are more important than the dispersal. And we also performed experiments in microcosms in the lab with more or less the same design as in the mesocosms. We tried different dispersal rates, trying to check their importance in the structuring of phytoplankton communities. So this is an outline of the design of the ECOGENE project and its main scientific question. And today I will present some information and some first inferences about the relative role of niche and neutral mechanisms with data from the 9 Aegean gulfs. We are still in the process of processing those data. So, first of all, some theory. There are mainly 2 approaches for the structuring of communities. The niche approach, which is based on competition between different species, and the selective role of abiotic factors on the growth of organisms. So for this theory, niche theory, environment mainly plays a role on the structuring of communities. For the neutral theory, which was proposed by Hubble in 2000, all species are ecologically equivalent. So the environment plays no role in the selection process, so competition between different species is not important, but mainly immigration and dispersal are the factors that influence structuring of communities. So, as I told you before, the main scientific question of our research in this project was to check which of those mechanisms is most important for the structuring of phytoplankton communities. Just have a look in our data of phytoplankton. As I told you before, we used 2 different approaches. In our knowledge, this is a complementary way to study phytoplankton: morphological identification and molecular identification. So we took the samples and use a part of the sample for molecular identification and a part of the sample for morphological identification. So we had 2, let's say, communities for each sample, one from each analysis. The morphological identification took place in an optical microscope, and the identification was up to the species level. We used 2 methods, one with sedimentation chambers and another one with filters, and then merged all this information to produce the community of the morphological identification. The molecular method was based on Illumina sequencing of partial 18S rRNA genes, and our communities look like what you can see in those 2 pies. On the left you can see an outline of the morphological communities. As you see, diatoms were the most dominant organisms, whereas in the molecular identification, dinoflagellates was the most abundant organisms. So there are differences between those two approaches. We didn't try up to now to combine this, to test this difference, but we only used those two approaches in a, we didn't try to compare the information from the one method and the other method, but we tried to use those two databases in a complementary way, more or less. A few words about the physical setting. As you see, the Aegean Sea has a very complicated pattern of circulation. You just get an idea from this slide. These are the results of a drifter experiment which was done in the past by Olson, and you see that there is a rather complicated pattern. Water inflows in the Aegean from the Dardanelles Strait here. This is water of low salinity and low temperature, and this water goes southwards near the island of Limnos. Its waters are characterized by very low salinities, and part of this water goes northwards. The water of Kavala and Thermaikos Gulf here near Thessaloniki are also waters of low salinity. Then salinity is higher here near Pagassitikos and Maliakos to the south. Salinity is rather high here in Saronikos Gulf near Athens. And as you see, waters of this high salinity flow northwards near Kaloni and Yera, which are two other gulfs that we sampled, where water is also of high salinity. So just keep that we have a very, very complicated pattern of circulation in the Aegean. Most of these points that I already mentioned can be also seen in those graphs. As you see, temperature is rather low in Kavala, Pagassitikos and Thermaikos, places that are here, mostly on the north, whereas salinity is rather low in Limnos here, which is very close to the outflow of waters from Dardanelles Strait, and Thermaikos also, and Kavala. Okay, so we have many differences in salinity, temperature, and the complicated circulation pattern. Considering nutrients, I have to say that nutrients are very important for the growth of phytoplankton and for the structuring of phytoplankton communities, for phytoplankton diversity. As you see, there are some gulfs which are characterized by rather high concentrations of nutrients, like nitrogen here, which is rather high, in Maliakos, Pagassitikos and Thermaikos, which are here along the mainland of Greece in the western Aegean, and rather low in the other gulfs which were studied. The same holds for phosphate, which are rather high in Thermaikos, and Yera and Kaloni here, or in those gulfs here, which are in Limnos and Pagassitikos. So more or less we also have a complicated distribution of nutrients rather complicated in space. So this is the whole setting of the environment considering physical variables and chemical variables in the Aegean. We tried to check which is the relation, which are the effects of those abiotic variables, both physical and chemical, on phytoplankton composition, as it comes from both the morphological analysis in the microscope and the molecular analysis. So for the data of the morphological analysis, you can see that there is a clustering in space regarding the composition. Here we have a cluster of those gulfs near the Limnos Island, influenced by the low salinity water that inflows from here. There is a cluster also here with Thermikos gulfs and so on. These are the gulfs of Kaloni in the island of Lesbos and Gera stations, another gulf in the island of Lesbos. So there is a clustering in space, and we can also identify some variables that are important for these spatial patterns, like temperature, as you see here. This graph comes from a redundancy analysis. Temperature is important. Phosphates are also important. Salinity. So it seems that abiotic variables which were considered, temperature, salinity, and nutrients, play roles on the structuring of phytoplankton communities. But because the whole setting is very complicated, we try to apply some more elaborate methods to identify patterns in space. So, more or less the same pattern was identified when the phylotypes were considered. We also have some clusters in space, like one cluster here with the stations of Limnos, another one here with the stations of Kaloni, and some variables which seem more important for the composition of phytoplankton when considering the molecular identification process. So there are differences, but there are also similarities, some clustering in space, and some important abiotic variables for this clustering. So, our aims of the aims of the work that I will present today. If we infer, first aim, if we can infer different patterns and processes when using morphotypes and phylotypes, the first aim. The second one, which processes are more important, spatial processes or local environmental processes in its gulf? And the third, what can we infer about the underlying mechanisms, which processes predominate, cis or neutral, in the structuring of phytoplankton in this complicated marine environment. In order to make some inferences about the scientific questions, we applied three methods in a complementary way, we think. First of all, a method proposed by Legendre and his colleagues, the method of principal coordinates of neighbor matrices, PCNM, which models the structuring of an ecosystem in various spatial scales. This method tries to identify all possible spatial scales in the structuring of a community, either fine or broad. And then those scales can be further used to identify the role of environmental variables or spatial variables and their possible overlapping. The second method is more or less basically the same like the first one, but has another very important feature for our approach. This method takes into account directionality in the identification of spatial structures. I have to note here that for us, for our ecological question, directionality is very important because it's a basic feature of the neutral theory. According to this theory, the neutral theory, dispersal processes are mainly important for the structuring of communities. So this method of asymmetric eigenvector maps takes into account directionality, the connectivity between our gulfs in this approach. And finally, we used another method which is called Generalized Dissimilarity Models, GDMs. This is a method proposed by Ferrier and his colleagues, and its most important feature is that it takes into account non-linearities in the effect of the various factors in the structuring of communities. Some information about the methods. First of all, for the PCNM method, if we have a regular transect of points, if this is our sampling design, points on a regular transect, equidistant points on a regular transect, this method produces a number of variables, some of them you can see here, either of broad scale or final scale, representing the possible structuring of our communities in space. Those variables are only related to our sampling design up to now. So for a regular transect, we get a bunch of variables like those one. If we sample on a regular grid in 2 dimensions, we get variables like those ones. Some of them are of broad scale and some of them are of fine scale, as you see here. And for an irregular sampling design like ours in the gulfs of the Aegean Sea, one variable is this one, which is graphed here. As you see, with different colors, we can see different values of one variable, one PCNM variable. So this variable that I have here in our sampling design represent more or less fine scale spatial structure. So if we start from our sampling design, from the location of our stations in space, we produce a number of those variables. This is the first step according to the method of PCNM by Legendre and his colleagues. Then we select a number of those variables which are correlated with our community. Okay, so we take a sample of this bunch of PCNM variables, only those that are correlated with our community data, if we have a community, or with the abundance of a single species if we study a single species in space. So, those selected variables represent spatial structures of our community. In our approach, the selected PCNM variables represent spatial structures which exist and are also shown from our phytoplankton community data. So we have two matrices with explanatory variables. Those PCNM variables, I'm talking about the selected ones, and another matrix with the environmental variables. For us, the environmental variables that we used were temperature, salinity, inorganic nitrogen, and phosphorus. So we have two matrices of explanatory variables and the response variable, which can be the abundances of the various species or binary information about presence and absence. And we can partition the variation of our community data into two parts, one environmental and one spatial. And we can also calculate the overlapping between them using a variation partitioning method that was introduced by Burkhardt. So this is the approach, and finally we have the amount of variation of our community that is explained by the spatial variables, by the environmental variables, the overlapping, and the residual variation. This is the PCNM method. We also used another method which is called asymmetric eigenvector maps, AEM, which is also proposed by the same research team of Legendre, which is almost similar with the previous one. We also calculate a number of variables according to our sampling design, to the coordinates of stations in space, but we also take here into account connectivity between stations. In order to take into account connectivity in our approach, how much connected are two stations between them, for example, how much connected are Kavala stations with the stations of Gera or Kaloni, or how much connected is Kavala with the stations of Limnos. We took the information from a hydrodynamic model which is run for the Aegean on a daily basis, and made a tracer experiment by letting a parcel of water in each gulf and trying to follow this parcel which was following the currents. And we performed this experiment through a number of simulations. It's a stochastic, it's a random walk algorithm procedure, and we managed to calculate the possibility of two sites to be connected and the weight of the connection. The weight has to do with the number of water parcels that started from one side and managed to arrive to the other side. So we had a matrix with all pairs of sites, the probability of connection, and the weight. And we used this information in the method of AEM of Legendre. And Two words about the third methodology, the Generalized Dissimilarity Models, the GDMs. We also have three matrices, one of them about the dissimilarity in phytoplankton composition, calculated on abundance or binary data, it's up to us. The geographical distance, which is a measure of spatial changes in phytoplankton composition and the community composition, and finally a third matrix with the abiotic information, the environmental factors. Here we used, as I said before, temperature, salinity, nitrogen and phosphorus. We try to incorporate something new in this method proposed by Ferrier. We used the variables that we found as significant from the previous methodologies into the GDMs method instead of geographical distance. So we ran the method with the geographical distance as a measure of separation in space, and then we used, instead of geographical distance, the PCNM and AEM variables, trying to define another scale of spatial resemblance, let's say, between stations instead of geographic distance. The outcome of a method like GDM is the role of each variable. I have an example here about the geographic distance. You can see that there is a change in community composition for distances up to 200 kilometers. So the rate of change is important here. And then there is no effect. So the characteristic of this GDM method is that we have we can calculate non-linear effects of variables on species composition, like the example that I present here. So let's have a look on our results and try to arrive in some conclusions. As you see, environment plays spatial variables, the PCNM variables, are more important than environment when looking at the morphotypes, at the morphological data. The same does not hold for phylotypes, for molecular data, where environment is more important. Another important thing is that the overlapping between environmental and spatial variables is not so important. This is very interesting because most of the methodologies that are applied when we try to find the relative effect of variables on a response, of explanatory variables on a response variable, are suffering from covariances and correlation. So this method manages to have a rather low overlapping. So there are some differences between the information that comes out from the morphological identification of phytoplankton and the molecular identification. So perhaps those differences have to be explored or used in a complementary way. It's not the same information. There are differences. Spatial variables are not so important for phylotypes, for the molecular information, but they are more important for in the morphological data. Among the variables that were used, salinity plays a key role in the morphotypes, and some spatial variables. But for phylotypes, as you see, mostly environment is significant. Space is not so significant. More information about the physical meaning of variables someone can derive looking at the RDA plots. Here I present one plot for the morphotypes, where we can see the relative role of the various variables, and when we try to resolve the physical meaning of those spatial variables, what is there behind, we can check the spatial distributions. For example, one of the variables that was important in our approach was PCNM4, for example, and we see that this variable expresses a change, mainly shows different values, mainly between two gulfs, the Saronikos Gulf and the Pagasidikos Gulf. So something happens with those gulfs, and we have a rather large effect of this variable in community structure. So each one has a distribution like this one, and if we want to study what is behind each one of those variables, we can study the distribution, the spatial distribution. Just a few words in this slide. As we remember that our main aim was to infer which processes are more important, neutral or niche. So the PCNM analysis that we already used finds spatial structures based on our sampling design, but it does not take into account directionality, connectivity between sites. This directionality is taken into account in the AEM analysis. So, by applying this AEM analysis, we found that space now loses its importance. So, if we take into account directionality, space has no importance. And we infer from that that the connectivity between gulfs is not so important. And the same holds for the molecular data here, on which more or less we found almost the same things in the PCNM analysis. Space was not important for the molecular data, both in the previous analysis and in this analysis. And the same thing we can see here when we see the important variables. As you see, the most important variables are the environmental variables when we take into account directionality. The AEM variables that are important are only of fine scale. So perhaps some fine scale processes are important, but for us the main scale that we were studying was the scale of the Aegean, and for this large scale of the Aegean Sea, directionality is not so important. Perhaps it's important for in fine scale. This can be resolved again by taking into account and studying those distributions of AEM variables. And finally, the results of the GDM analysis, as I told you already, we used all those approaches rather in a complementary way, trying to see if the results of each analysis are also produced by the other. And we can see here that the proportion of deviance that is explained, or the proportion of variation that is explained by this model when geographical distance or PCNM variables are used is rather high for morphological data, and it's rather low when AEM variables or directionality, remember, is included. So it seems that when you take into account connectivity and directionality, the variation of phytoplankton communities cannot be resolved better. And almost the same, more or less, happens with the molecular data. Here, when we include geographic distance, we have a rather high explanation of variation, which drops when we use PCNMs or AEMs. But remember that when we applied those methods for the molecular data, space was not so important in any case. So our main conclusion comes from this drop between the 40 something and the 27, and we believe that connectivity is not important after all for the structuring of phytoplankton communities in the Aegean. Yes, I'm finishing. When we consider the role of the various variables, I have an example here for geographic distance. As you see, geographic distance is just important up to a distance of 200 kilometers for the Aegean, and then there is no difference in the community variation. But for molecular data, this variation still increases, and perhaps this is related to the higher effort that is put in the identification of organisms when you use molecular information. The abiotic variables have also effects on the community composition, and you see that there are different slopes across the ranges. So some variables are more important in high values, like the salinity, temperature. Also, his effect on community structure is rather higher in the higher values, and so on. Or for phosphorus, for example, its effect is more important in low values, and so on. And trying to arrive in some conclusions and some future perspectives in our work, we have shown that different methodologies are very useful when you try to arrive in a conclusion in your scientific question. So it's very important to use different methodologies, but in a clearly stated complementary way, according to your questions. Those methods are, this complementarity of methods is extremely useful in complicated environments like the marine environment or the Aegean environment. Morphological and molecular diversities indicate similar, more or less, drivers. But there are differences. So we have to further study those differences trying to arrive in some conclusions by always looking in complementarities in those data and not making comparisons. It seems that mainly environmental factors, and namely temperature and salinity, physical factors, are the drivers of phytoplankton diversity in our case. But inside this spatial component of PCNM and AEM variables, or inside the residuals, there is some hidden information which we have to investigate, because the residual variation was rather high, and we have to see what is there. It seems that local environmental processes are more important, and when we try to define a scale of separation in space, mostly geographical distance is important. And connectivity and dispersal mechanisms seem to be of low importance in our case. And from this conclusion, I arrived to the final conclusion, and the main aim of our work. It seems that niche processes mainly structure phytoplankton and not neutral. Of course, we have to further explore the information that I presented here, and we also have to consider the information that we gathered from our experiments in mesocosms and microcosms, where conditions were set, and perhaps it's easier to arrive to some conclusions when you study controlled information from controlled experiments. Thank you very much for your attention, and of course I would like to thank all the people that worked in ECOGENE, and finally the MASTS for its important support to be here with you and talk about our work in the Aegean. So thank you very much.
Thank you, George. Because this is broadcasted as a webinar for people in the MASTS community, I think it might be best if we give them the opportunity to ask some questions beforehand. George is going to be around all day, and he's also coming to the pub afterwards, so we can ask as many questions as we can after this. John, do you have any questions for George?
Yeah, Sophie, we have one question from Paul Tett. Paul's asking, have you been able to define niche for characteristic species from your data?
Please repeat, John, I didn't hear. If we were able to define what?
Have you been able to define niche for characteristic species from your data?
No, we didn't work so far with characteristic species. We just work with the whole community, if it is the question. But we plan to apply those methods in the future for certain species.
Okay, thanks.
Is it okay?
Yeah, yeah.
Someone can apply the same methods for one species, for just one species, either for its abundance or its presence absence, as they are applied for the whole community. So we have already identified some groups of organisms that behave in the same way, and we plan to apply these methods or similar methods to groups of organisms or to certain organisms to find what is happening there.
Okay, thanks.
John, do you have any other questions?
No, not at the moment. I don't know if you want to go back to the room, Sophie, for some questions to give the online audience a chance to type any more in.
Okay. Okay, thank you very much. So we can take any questions from here, if you would like to ask something. Okay. So thank you very much all for coming, and thank you, George, for...
Thank you very much. It was my pleasure to be here and to present our work. And I hope in the future we will have
more opportunities.
More opportunities, yes.
Okay, thank you, everybody.